Okay, in our last segment, looking at antibodies, antigens and antibodies, we're going to take a look at how antibodies can be used as anti-cancer agents. Now, we will look at their ability to be used in vaccinations and in ability to protect you from um, infectious diseases, toxins, and things later when we cover those concepts looking at vaccines later in the course. For now, we're going to take a look at how antibodies can be used as anti-cancer agents. And one of the things we want to understand is that, and what you should be fully aware of, is that this is an incredibly growing field, cancer immunotherapy. There are 30 or 40 clinical trials going on, if not more, worldwide in terms of their use. A number of antibodies have been developed as anti-cancer agents, and we're going to kind of take a look at that a bit in the in-class exercise that we're going to do this week. Um, but this is a really growing field, and cancer immunotherapy is really red hot in terms of drug design and drug development right now. Um, Cancers, as the slide says, human cancer actually is pretty immunogenic. You actually do generate um, immune response to antibodies, to cancer, through some of the overexpressed antigens that tumors can have on them. There are many of these, and just an example, um, this is an example. Some of these are like PSA for prostate cancer, PAP, mucin, CA for colon, HER2 for breast. There's a number of these types, and you can kind of look them up and tell which ones they're associated with. But there are many, and there's been a real effort over the last 20, 30 years to try to, particularly last 30, maybe 40 years actually, to try to understand if we could find antigens that were relatively specific or at least very well overexpressed on cancer cells. Then we could use, as Paul Ehrlich hypothesized back at the turn of the 1900s, development of a magic bullet that would go after cancer. In his case, he was talking about infectious diseases, but it turns out that there are a number of antigens that can be potentially used for that. And if you look here, this graph is interesting because this is the percent of colorectal patients with an antigen called HER2/neu antibodies. So these are patients that have developed antibodies to this antigen that's particularly associated with colon cancer, as well as breast cancer. So if you look at this, here's normal. If we take population of 200, we'd see no antibodies to this antigen, HER2/neu, okay? HER2. If we look at all colorectal cancer patients, interestingly enough, 14% of them actually have antibodies have been shown, you can see antibodies to HER2/neu. If we look at patients that are HER2/neu positive, in other words, their tumors express this antigen, and so in this case it would be 13. Almost half of them actually have expressed antibodies to the HER2/neu. And if we look at patients in which their tumors don't express this antigen, and that is true in some cancers, even though they're the same cancer, they may be deficient in the antigen. Almost none, like only 5% actually produce antibodies to HER2/neu, probably meaning that at one point they did have expressed this antigen, but have gotten rid of it. And so it's true that tumors can evolve and eliminate things that or suppress the immune response so that they can eliminate the potential for antibody development. And so this shows us that the immune response can actually generate there can be an immune response to cancers. This is just one way. We'll talk about other ways that's going to be the case too. But the point here is that there are antigens to tumors, and you can't, and there is a development, interesting enough, that people who are cancer patients actually do develop some of these antibodies. So the use of monoclonal antibodies can be in three different areas. We talked about monoclonal antibodies before. They bind one antigen and determinant. You can use them for diagnosis. For example, using them to see if the tumor has that antigen on its surface by using diagnostic means, a variety of different diagnostic means. You can use it to monitor disease progression. You might be able to use PET imaging, for example, to monitor where the disease is by using an antibody, by radiolabeling that antibody, or where it and to the extent that it's potentially there, that specific disease is there, cancer is there, and you can also use them for therapy as we'll see. So I'm going to introduce a couple of these just as concepts right now, and concepts of how this works and their success in this way. And we're not going to cover this in exhaustive detail, just a couple antibodies to show you how this works. And then in the exercise this week, you'll get a little bit more taste of this. So one of the first ones that was approved way back in the late 1990s was rituximab. This binds to an antigen which is referred to as CD20. That's the cluster. CD is just a standard nomenclature for a cell surface antigen. And so for B cells, these are on B cells and B cell malignancies, you'll find CD20 can be expressed and potentially overexpressed on those cancers, and it's also expressed on B cells. And so this antigen CD20 is expressed on it, and the antigen has been found to be a calcium channel. It can be phosphorylated and it can be activated by serine tyrosine kinases. It's the upregulation of adhesion proteins and c-Myc, so it can, and this being an oncogene, and it's expressed most interestingly on B cells and B cell malignancies. And so an antibody was developed, actually a monoclonal antibody was developed, this rituximab to this, and its mechanism of action is as it stimulates the apoptosis by calcium flux induction. So it does, when it binds, it can stimulate the cells to sort of go into apoptosis. That can happen. It has really a major part is it induces ADCC, which we've talked about, antibody directed cellular cytotoxicity. And so in that mechanism, and it can also mediate complement fixation. So if it binds to cells, it can, and you have enough of that. Now remember, you need enough of those antibodies to bind to the cell. It will initiate complement on the cell surface. So it has two, maybe three different mechanisms of activating cell killing. Here's a CD expression, and these are the different types of B cell leukemias or B cell leukemias that, except for CLL, different types of these leukemias. And what you'll notice is that rituximab can be used in a variety of these, and you don't have to memorize these. It's just evidence that there's a variety of B cell leukemias and lymphomas, and that express CD20. So as a consequence, rituximab can be used against these different kinds of B cell malignancies. Now, when it was used in one of its early trials with about 39 patients, and they were looked at over about 12 months period. This graph is a graph which shows you the 100% of the patients, and this is the percent that's progression-free survival. So they survived, and they did not have the cancer over this period of 12 months. And you can see over this period there were 20% of the patients died, However, 65% of the patients had an actually overall response when you look at this. Um, and so 65% had overall response. That means they responded either partially or completely. 27% had complete responses. In other words, they appeared to be completely cancer-free. And 38% had partial responses in which either some or part of the cancer appeared to be eliminated, and it appeared to be eliminated in their, but not all of it. So that's pretty good. These kinds of response values are quite amazing, because that means you have like about 20 to 30% of the patients being potentially cured. If you were to look at it, and this has gotten better with in the newer developments, but if you were to look at um, just a normal anti-cancer drug, you may get as high as, if you can affect, 10 to 15% of the patients in which you might get this kind of result, that's really excellent. That's really good. And so you can see here, this is almost double what you would get for potentially for a known, sort of an typical anti-cancer drug, small molecule drug. So that's one of the reasons for a lot of excitement in these kinds of drugs. Now, rituximab is another one. It's quite popular. This targets the HER2/neu, which we sort of talked about before. It's a growth factor. It's a very important growth factor, and it's a membrane-bound tyrosine kinase. It's involved in cell growth and differentiation, and when it gets stimulated, it can upregulate. It can really lead to progression of the disease. And the higher amount of HER2 you find on the breast cancer cells, the more prone they are, the more likely they are to become potentially much more dangerous and metastatic. They bind to the epithelial growth factor, which is a factor that leads to growth, and can be upregulated in 20% of breast cancer patients. You'll see this, and some patients will have this upregulated, and it can control that cancer. It's also overexpressed, as we saw. There's some overexpression in colon, but it's also is overexpressed in ovarian and stomach cancers, and so there's a use of potentially this as a target for those cancers as well. Now, natuzumab's mechanism of action is a little bit more complex. It can down-regulate the HER2 expression and therefore inhibit the proliferation of HER2, so blocking a key pathway, which is the signal transduction AKT pathway. And so it can block this pathway, and therefore we might tune down its ability to the cells to grow. It also can undergo ADCC, and so it results in ADCC, attack of the immune cells against the cancer cells. And it's also shown to have, it's quite an interesting antibody because it also can downregulate angiogenesis. Angiogenesis is the process used for tumors in which they need to form their blood vessels. To form tumors, as you form tumors, they need blood. They need oxygen, they need nutrients, and so blood vessel formation in them is really critical. So if you can stop that, you can, in theory, stop the spread of that tumor, the growth of that tumor, and potentially lead to its death. And so rituximab has been shown to have an ability to downregulate this process called angiogenesis. So all of these things are contribute. As far as immune interaction, though, ADCC is an effective way to go after these cancer cells. Now, if we look at some of the clinical data, this is an early example, early clinical study, in which you can see the number of, out of 200 patients, the number of patients with overall response was about 15%. Complete responses was 8 responses, partial responses 26. 12 patients had minor responses, and 47% number of the disease patients had stable disease over greater than 6 months. The median duration of the response was about 9 months, and the median time to disease progression was 3. So they're stable, sort of median, and to for these patients with metastatic cancers, metastatic breast cancer. So this would be the worst kind, one of the worst kinds, was great, over 3 months, and this median survival was 13 months. So this turned out to be pretty effective because there really wasn't much for the metastatic breast cancer patients, and this result was very much superior to anything that was out there. And you can see here in this pivotal phase 3 trial, if we look at this phase 3 trial, what we see here is three different graphs. One in which you have trastuzumab, which in this case would be, it has another name referred to as Herceptin, but trastuzumab in chemotherapy, and the time progression was in this phase 3 trial was 7.5 months, whereas if they received just chemotherapy, it was only 4.5. It was 4.5 months. So you got an increase of about 3 months there of stable disease. For these, response rate, the percentage with trastuzumab and chemotherapy was 50%, whereas with just chemotherapy it was only around 30%, so an increase of 20%. And the percent live after 1 year survival with Herceptin or trastuzumab and chemotherapy, it was roughly a little bit, it was about 80%. It's about 70, it's actually 77%, 80%, whereas with just chemotherapy, it was roughly around 70%, a little lower than 70%. And so you got an increase of roughly about 10% there in survival. So clearly trastuzumab has a good effect, and it's been one of the one that's been used and has been quite successful against metastatic breast cancer. Quite successful in the sense that this is a really hard cancer to treat, and it's had success against that. Its efficacy, it has reduced recurrence by 50% for patients who have been treated successfully. It increases the survival, has increased survival rates by 30 to 45%, and it does have a major toxicity, but this major toxicity is reversible, and it does have, because it appears that HER2/neu is expressed also cardiac tissue, and so there has been some cardiac issues with cardiac dysfunction related to that, but that's reversible once this therapy has been stopped. So I know that's just kind of a short section. We're going to look at some of these in the in-class in terms of looking at the types, some antibodies that have been FDA approved recently, and to give you, that gives you a taste of the antibodies their use as anti-cancer agents. Now here's some review questions. There's 3 here that you can take a look at and answer, well, I'll be stopping here, so you want to stop the lecture here, that's fine, and the next slide will be the answers.